So we know that there are multiple types of membrane proteins, and we have both integral membrane proteins that span the whole membrane or just one of the leaflets, and then we have peripheral proteins that kind of weakly associate with the membrane from the cytoplasmic sites, but in the middle and kind of in between those two classes of membrane lipids, we have lipid-linked or anchored membrane proteins. And those types of membrane proteins are covalently linked to some hydrophobic fragment that helps them to anchor into the membrane. So that hydrophobic fragment actually either a fatty acid or an isoprenoid polymer, as you can see here. And correspondingly, we have prenylated proteins and fatty acylated proteins. And for prenylation, we have either farnesyl residues, in which we have like those 5 carbons, like that, right? Those are units, isoprenoid units. And for farnesylated proteins, we have 3 units. And so in this case, n equals 2, because I kind of have one more unit right there. Or we have geranyl-geranylated proteins, in which we have 4 of the isoprenoid units, and here n will be equal 3. The attachment for those isoprenoid units will be through the cysteine residue, and pretty much I will need to erase that hydrogen, and I will need to show that here I have that cysteine residue, which becomes a terminal residue of that protein, C-terminal residue of the protein. And the linkage here is thioether. For the second group of lipid-linked membrane proteins, we have 2 types of fatty acids that can be attached to the protein. And depending on the type of fatty acids, the attachment and the type of amino acid side chain to which it attaches would be different. So here we deal with myristic acid that has 14 carbon atoms. So we call it like that, 14 carbon atoms and no double bonds. So those proteins are called myristylated, right? Myristylated, but the fatty acid is called myristic. And the attachment here will be via amide linkage. So I will have to erase over there, and I will have to put an amino group, and that amino group will come from the N-terminal glycine residue. Right? So here we have cysteine, right? And it becomes, after processing, the C-terminal cysteine residue. And in case of myristylation, we have N-terminal glycine to which that fatty acid is attached via amide linkage. And finally, a little bit longer fatty acid, the one that has 16 carbon atoms, we call it stearic acid, palmitic, of course, palmitic acid. Even though it's a similar hydrophobic tail, but the attachment here is via thioester group, and you have a cysteine residue right there that is that can be in the middle of the protein polypeptide chain, but of course it should be exposed to the, it should be a surface amino acid side chain, because in the middle, in the core of that globular protein, of course, we will not have that palmitic acid. It will not access the core. So we have cysteine, and the linkage here is thioester. So that is the difference. And because of the chemistries of each linkage, we can expect that this kind of modification is more or less transient, right? So it's much easier to break the ester bond than amide bond, for example, right? And so you can envision that some enzymes that may catalyze hydrolysis of that bond, and that would liberate the protein, and it will no longer be anchored to the membrane. But for other types of chemistries, prenylated proteins or myristylated proteins, it's not as easy. So they are more of the permanent kind of modifications. So that would affect, of course, the functions of those membrane proteins. There is one more lipid-linked membrane protein, which is called GPI modified, and that GPI stands for glycosyl phosphatidyl and I stands for inositol. So we know that phosphatidyl inositol will be that lipid that allows the protein to get anchored at the membrane, but it will be also glycosylated, and in fact it has more than one sugar residues attached to that. And because we have like a bigger oligosaccharide fragment attached, kind of serving as a linker pretty much between the protein and phosphatidyl inositol, we expect that chemistry and that arrangement to be projected toward the outside of the cell. Are types of lipid-linked membrane protein that have so different chemistries and correspondingly functions.